Hi everyone, my name is Ioana. I am a PhD student at the University of Bristol. I'm studying engineering mathematics. Thank you very much for having me today to speak to you about how we can minimize an E. coli cell, actually reduce the size of an E. coli cell using machine learning and whole-cell modeling. So I would like to start by telling you what a whole-cell model is. I know that there was a talk about this before, but actually the whole-cell model that I'm using is a bit different. So generally speaking, a whole-cell model is just a mathematical model that should include everything that happens in the cell, meaning all the molecules, the whole cell cycle, all the genes, all the processes. Up to now, they've generally been dynamical models with some stochastic elements. So the whole-cell model that I'm using is the E. coli whole-cell model published by the Covert group at Stanford University in 2020. So it's a model that includes all these processes here and their interactions. It has over 10,000 equations and over 19,000 parameters. The version that I'm using here, it describes the functionality of 1,219 genes. And we can simulate different environmental conditions. And what is really nice is that we can simulate gene knockouts, both single and multiple gene knockouts, which is quite important for our task. So before going into how we managed to reduce the genome, I just wanted to show you an example of how what the output of the whole-cell model looks like. So first of all, we can simulate several generations. So several generations of cell division. So here, let's say we start with generation 1. We have one cell. It takes around 15 minutes to produce the output of this simulation on a typical desktop computer, and it outputs 200,000 time series. If everything goes right with this, the cell divides. It has two daughters. Each of them in turn have 200,000 time series, and it takes 15 minutes to produce them, and so on. So you can see that as we increase the number of generations, the number of cells that we simulate grows exponentially. So one of the first things that we had to decide was how many generations we want to run, because in order to understand if a cell is healthy or not, or if a genome is healthy or not, we would have to run a few generations to understand if the population of daughter cells behaves as expected. So after some investigation, I understood that we should simulate at least 6 generations. So in 6 generations, we start seeing cells that do not divide. Another decision that we made was that what we mean by a healthy genome is so a genome is healthy if it produces dividing cells in 6 generations. So all the cells in 6 generations divide as expected. So already, I guess you can kind of think that we would have to run many such simulations to produce a minimal genome, right? Like if we start removing cells until we find a genome that divides, we'll have to run many simulations. So, and this is not really possible with the whole-cell model the way it was, especially if we want to run 6 generations. So I thought, okay, what if I find a way to bypass all these simulations, but still be able to predict whether all cells in 6 generations divide? So what I did is I trained a machine learning algorithm, a classifier, that based on the information at the end of generation 1, is able to predict whether all the cells here divide or not. The idea being that if this works, we will only have to run 1 generation, but still be able to predict the phenotype that we're interested in. So this model actually worked quite well. We had an F1 score of above 92%. This is a way to measure accuracy that kind of takes into consideration both the false positive and the false negative. So it works when the data is unbalanced. So like if we have more cells that don't die and less cells that die, which is the case here. And we were also able to look into what the classifier does. So here, these are the 4 features that we use, and we see that when the cell mass is high, for example, that is likely to, the algorithm is likely to predict that the cells divide successfully in 6 generations. If this is lower, it's likely to predict that they won't divide, which kind of makes sense biologically. Like if there's not enough stuff there, the cell will probably fail to divide. So, okay, now that we have this, we are actually able to to look for our reduced genome. So we are able to run many simulations and hopefully find a smaller E. coli genome. So the first step to do this was to run all the single gene knockouts. Once we have all the single gene knockouts, we select only the non-essential genes, which are the ones that for which all the cells in 6 generations divide. Once we have this list of non-essential genes, we create 56 segments representing different percentages of these non-essential genes, and we simulate the cell using the whole-cell model, removing each of these segments one at a time. Once we run all these simulations, we take the highest segments from stage 2, for which when deleted, the cells divided successfully, and we add all the non-overlapping segments for which the cells divided successfully, and we create a power set. We simulate again, and you can see already here that by now we would run above 10,000 simulations, because here we have like 1,200 each single gene knockout, then we have 56 here, and then running all these combinations, it's easily like around 10,000. So that's why having this machine learning surrogate, which can emulate the behavior of the whole-cell model, is actually quite useful. So the next step is we take again the largest segment that we deleted here, and that still produced a dividing cell, and we split the remaining genes into 8 groups. And again, we combine this largest deleted segment with these genes. We simulate, and basically, if some of these simulations work, so the cells divide, we just update this largest deleted set. If none of them divide, it means basically that these segments that we're appending are too big, so we need to append less genes. So we just append 1 gene at a time, and based on the results, we update this list of remaining genes. I can explain this in more details if you want separately, but it's basically a loop that keeps going until we have less than 16 remaining genes. So after we finished running all the simulations, which were around 20,000, we obtained a reduced genome. So from 1,219 genes in the whole-cell model, we reduced it to 742. And because of the richness of output from the whole-cell model, we were able to look at different macromolecular masses and their corresponding time series. So what we see here is that the reduced genome has lower cell mass and lower growth rate. But interestingly, it has higher protein mass and higher RNA mass. And this is something that we are still kind of, we're thinking and trying to understand whether this is something biological, so whether some proteins are produced in excess, or is it something something that the whole-cell model does in order to keep the cell alive. So to keep the cell alive, it tries to produce more proteins. So in conclusion, we managed to speed up the whole-cel model simulations from approximately 38 years to 2. This is on a standard desktop computer. Of course, we use a supercomputer, so it took us less than 2 years to run the 20,000 simulations. But even so, without this, I don't think we would have been able to do it even on a supercomputer because of the multiple generations. It kind of has to wait for one generation to finish before it can start the next. The genome was reduced from 1,219 modeled in silico genes to 742. And because of the richness of the output of the whole-cel model and the explainability of the machine learning model, we can understand the patterns that cause failure in cell division. And we can investigate the time series of both the reduced genome and a wild-type cell. Thank you very much. I'd like to, thank you for allowing me to speak. I'd like to acknowledge my supervisors and my collaborators, both in Bristol and at Stanford. And my funding body, PSRC, and Oracle for allowing us to run some simulations on their clusters. Thank you. Are there any questions?
Before I go, anyone else want to take a shot? Ioana, so I don't believe, I'm sure you don't believe that you can make a viable E. coli with 742 proteins.
Yes, so I think there are a few things here. First, the first question, I guess, is the 1,219 viable? Because the whole-cel model at the moment only contains 1,219. So I don't think it is. I think it is just what, it's just the model works with this because it was adjusted such that it works given the experimental data available. But I think what this tells us, which is why I don't call it a minimal genome really, because I don't think it would work. But I think the message of all of this is that maybe some of these groups of genes that we delete using this approach could be useful to guide future experimental deletions in E. coli. Because when I compare it to the reduced minimal E. coli genomes from the literature, there isn't much overlap. So I think the smallest that I found had like 56 segments of genes deleted. Whilst with this approach, we are able to look in more places across the genome. So what I would like for this to kind of suggest is that perhaps some of these segments that we suggest here could be tested experimentally and could guide us towards an even smaller genome.
In a decade ago, a little more, we were trying to make a minimized E. coli. And this was a project funded by Novartis Vaccines and Diagnostics. They were hoping that we would be able to build a minimized cell that produced extra OMVs, outer membrane vesicles. And we were deleting clusters of genes based on what we knew. And we found that in some cases it worked great, but sometimes the more we took out, the effect of taking out a gene would be idiosyncratic, or taking out a group of genes. So we were using recombinering to remove genes, and we would take out 4 or 5 genes, and suddenly recombinering stopped for no reason that we understood. Mm-hmm. And your model doesn't consider any genes that you don't know the functions of.
No.
How do you deal with the potential of synthetic lethals? Do you know that term?
I heard of it, but I'm not sure I remember now.
So a synthetic lethal is two genes that perform, two non-essential genes that perform the same essential function. And so you can always take out one of them.
But
imagine you've got a jet airplane, and neither engine is essential.
Yeah.
But you don't want to be on the plane where both engines go out.
So why would that not be included in the model?
Well, I'm sure that there are annotated genes that you have two genes that perform the same function.
Yeah.
So in your model, do you consider things like that?
Yeah. So when I analyze the genes that I delete, I see that I delete some gene ontology categories, which can be deleted because there's another gene that performs that function. To be honest, I haven't tried to delete both, so that category and the gene that performs that function, but I guess I could try that. But it looks like it does take that into consideration. Yeah.
Is there a way you could go through what we did to build SYN3, our minimal cell, to see if where we have a much higher fraction of the genes annotated? So with E. coli, you've got about, you have 1,200 out of 4,000 genes are in your model.
Yeah.
Whereas in ours, we have 470 genes, and we know what 390 of them do.
Yeah.
I know it would be a massive building project, but.
Well, so I think in Stanford, they are trying to add more and more genes. So this is an ongoing project. So actually the version that we use here has 1,200. I think the latest version has maybe 1,400. So I hope there will be, in a few years, a more complete whole-cell model. Yeah, I don't know if that answers your question.
Yes, it does. Thank you.